Okay, so it looks like recording. So like I said, I'm going to get these recorded and put up this weekend. Heads up, let me put this in the chat just in case someone comes in and they see it. We're going to have our first quiz next Wednesday. I got you. Oh, get this posted. Okie dokie. Okay. So I want to ask your opinions. Okay, because I would like to treat this as face-to-face as possible. Do y'all think it would be odd if I put the quiz up and then we had us Zoom so that I can be there to, one, watch you, two, answer questions?
I think that would be fine.
Be fine. Okay. Okay. So that way, because what I would like to do would make it very much like a quiz in person. Moving forward, I'm going to make sure this one doesn't have any like drawing or anything. But do any of y'all have a what do you call it? Abilities to print at home?
I can print.
Is there anybody that can't print? Okay, and actually, it's not really a necessity as long as you have some paper, because there's going to be some stuff I think on the quizzes that I think would be really useful to have where y'all can actually draw it out, much easier than trying to type out something. So one thing you could do is I think what I'm going to do is I'm going to post the quiz as a what do you call it, like a Word document, and then you can type your answers directly into it. And then if there's something you have to draw, a diagram, make a table or something, you don't really have to even print. You could just take a piece of paper, put the number of the question, do that by hand, and then we'll take a picture and upload it that way. So we're gonna try this out. This might be more trouble than it's worth, but I've not tried it before, but I think it might be useful. So, okay, hopefully y'all are okay with that. Sounds like most of you are. We'll see after Wednesday how that goes. Okay, let's get into sharing screen. And we're gonna pick up with mitosis. We are not going into great detail, but I'm just reminding you of some things that are basic biology and reference to plants. So mitosis refers to the process of cellular division that produces two daughter cells. And of course, we get equal amounts of DNA. How do we get equal amounts of DNA? Well, the DNA has to get replicated in our S phase. And when it gets replicated completely, we can divide it during mitosis and separate it, and the cell will get completely two sets of copies, one for one cell, one for the other. This occurs in meristems. Okay, meristems, 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 and I'll say it again, meristems. Can anybody guess why I emphasize that word? Real simple. Is it important?
It's important on the quiz.
You might see it somewhere again. Exactly. I can't promise it's going to be on the quiz per se, but it will show up again. And so where mitosis happens, this is really important. We talked about the palm tree and how, you know, if you've only got one meristem and it gets destroyed, bye-bye growth for that organism. Okay, we'll hit that again as we talk about this a little more. And I'll tell you, if you ever take plant physiology with me, you will note that meristems become exceedingly important as we talk about the growth development of a plant. But you may think, well, what if I don't take botany? We just don't take plant phys, man. I'm just stop with botany. Well, if you ever grow any bushes and you prune those bushes, you're actually pruning meristems. You're shaping that plant by cutting off some meristems and allowing other ones to activate. So it actually does have a relevance to you. So if you grow anything like that, or if you top a tree, you know what I mean when I say top a tree? You know, you prune the tree and it gives it kind of a nice rounded shape. That's something that deals with meristem growth. Okay, in mitosis, which happens in the meristems, and there are some other places as well. We'll get into that. But the majority of this is going to happen in the meristems, and it's going to be in 4 phases. You are very familiar with all of these, I'm sure. Prophase, this is where the chromosomes appear. They'll condense, and you can actually see them. We will look at some prepared slides of chromosomes under the microscope in lab. Metaphase is where they line up in the middle and they form sister chromatids. Okay, does this sound familiar to everybody? Okay, this is good. If it doesn't sound familiar, we need to pull out a Bio 1 refresher on this, because this is something that we want to make sure you really got. Anaphase is where they start to separate, so they're actually getting pulled apart. So metaphase puts them together. Anaphase is when they start to get pulled apart. And for the most part, plants and animals do this the same way. In telophase, things get a little different for plants. So we get the daughter chromosomes surrounded by a nuclear envelope. So they get to the other side where the 2 cells are going to end up becoming. They get surrounded by an envelope. The daughter chromosomes become longer and thinner. You see nucleoli. Don't know why I can't talk this morning. Appearing. And the spindle fibers that are pulling them apart disintegrate. And then it's in this moment, cell plate, where you see probably the biggest difference for mitosis. The term cytokinesis. By the way, does anybody know what cyto stands for? What is the prefix cyto? If you're studying cytology, cell, exactly. So this is cytokinesis. This is basically like the step that's going to make these 2 completely separate cells. So we see this forming from Golgi vesicles that will line up, and they form what's called a cell plate. What's probably one of the most major differences when you look at a plant cell versus an animal cell?
Cell wall.
Cell walls, okay? And this causes a problem for a plant cell versus an animal cell. In animal cells, when we do cytokinesis, what happens? Does anybody know? I should say, anybody remember? They don't do a cell plate. They do something else. Nobody? How do 2 cells become, sorry, 1 cell becomes 2 at the end of mitosis in animal cells? Do they just magically go, bam, they're into 2? Okay, so you've got an animal cell. Notice I'm making it round. And what happens is we get something called a cleavage furrow that causes an indentation, so you end up something like this. Okay, a cleavage furrow is where we take that first cell and it kind of gets squeezed, and it's going to get squeezed into separating it into two. Okay, why can't you do that with an animal cell? Brandy, I'm going to have you answer again. Same answer.
Cell wall.
Okay, so why can't we do this with a cell wall?
It can't be pinched.
Exactly, it can't be pinched. So it's a more solid object. It's not squishy, soft cell membrane. There is cell membrane, but the cell wall is in the way. So we form a new cell wall in between the two along what's called the cell plate. And so here we go. We've got prophase. We have metaphase where they all line up in the middle. So condensing, lining up, anaphase where they get pulled apart, telophase where they're actually separated and a cell plate forms. Cytokinesis is part of this last step of telophase. As that cell plate forms with those Golgi, we will continue the process until two cell walls are actually fully formed and we have two cells. Okay, so this is the major difference between animal and plant mitosis. Well, I would say it's the main difference. There's probably some other ones, but the more obvious visual difference in how they work. You can't pinch the cells apart. You actually have to, and remember that's a cleavage furrow. Here we have to create a cell plate and kind of put up something that we can build two cell walls that will separate these new cells apart. Now, do me a favor. I would like you to maybe make a mark or note or something that when we get into algae, I would like you to come back to this slide, okay? Because there's going to be some differences, a little difference in how algae does this last step of telophase, the way they line up everything. And I just want you to have a reference to come back to. Okay, we're going to briefly hit endosymbiotic theory, even though we really have already covered it. So we originally think that we had a eukaryotic cell that engulfed a bacterium. Now we've got two membranes around this cell, and one of those sets of membranes becomes the outer membrane and another one becomes the inner membrane. Does anybody remember what those inner membranes are called in the mitochondrion? Starts with a C. Nobody? They're the cristae. Okay. You might want to start reading over your notes. Okay, so what happens is this engulfing probably happened twice in the case of plants. So we start off with an ancestral prokaryote, no internal membranes. Internal membranes start to become possible with infolding. We get a nucleus formed from that internal membrane system. And then we engulf that purple bacterium that can do aerobic respiration. And we end up with what's called a mitochondrion. And this is our ancestral eukaryote, okay? Now, why do all eukaryotes have mitochondria, but not all eukaryotes have chloroplasts? Well, the mitochondrion was probably absorbed first, and that increased its ability to be able to produce energy for itself, okay? And then over time, some of those cells developed a chloroplast by engulfing some kind of cyanobacterium, okay? And we end up with two endosymbiotic events in this lineage. Everybody good with that? Okay. So basically one thing we noticed is that instead of getting destroyed or consumed in some way, the endosymbiont is persisting and it doesn't harm the host. It actually becomes a benefit to the host. The evidence for this is that we've mentioned some of these things. Do you remember the evidence for endosymbiotic theory? What are some things that mitochondria and chloroplasts have that make us think, probably this is the case? There's like 2 or 3 things you could mention. What's an evidence when you look at it? Okay, double membrane, absolutely. What else is inside? You can even look at the picture. That's DNA, exactly. Now, here's something interesting. How is DNA different in a eukaryote and a prokaryote? What format does the prokaryote have? Like a bacterium, what's their DNA look like? Are they in big long strands? Actually, I kind of tricked you, Elena. I'm sorry. They're not in big long strands. They're actually circular. Okay, Brandy, what were you saying?
I was gonna say they're coiled.
Yeah, yeah. So you can see that actually in the picture, can't you? So basically, it's just one big circle, and they get twisted up.
So
Good job. Yeah, sorry, Elena, for tricking you. Um, I will do that from time to time. Not just you, but everybody else too. Um, so, and this is something interesting. Guess what? When you look at the bacteria inside of uh, eukaryotes, mitochondria, or chloroplasts, they're circular DNA, just like you would get in a bacterium. Whereas in a nucleus, you get these big long strands, multiple strands we call chromosomes. So you don't get multiple chromosomes in bacteria. You get one big circle piece of DNA. They also can produce their own ribosomes and proteins from those ribosomes. So they have cellular machinery to be able to potentially keep themselves fully alive. One problem with this is that they cannot be independent anymore because some of the genes that are in those in that DNA have been moved from the mitochondria to the nucleus. I don't know about y'all, but that's fascinating to me that DNA actually has that opportunity to move. Okay. I want to tell y'all all kinds of stuff that's not relevant right now. Um, Random thought. Do you know that there's a plant out there that can actually like give its chloroplast to other plants? And there's actually an animal out there called the sea slug that can absorb chloroplasts and become photosynthetic. How cool is that? An animal that's photosynthetic because it steals chloroplasts from algae. Just a little side note, kind of fun stuff in this cool world. Let's try not to kill it all before we get to discover it. Okay, so the mitochondria are derived from purple bacteria that use oxygen to make ATP, so aerobic respiration. And as I said, the endosymbiont loses the ability to live independently from the host. And some of those genes actually get transferred to the nucleus of the host cell. So if you take out a chloroplast, it doesn't have everything it needs. The host actually is now literally doing the word host. It's hosting these by doing some of the processes using some of the DNA that the mitochondrial chloroplasts would normally have been able to do on their own. Now, chloroplasts are really cool. They're bigger, as we talked about. The endosymbiont is probably an oxygenic photoautotroph, maybe a cyanobacterium. Um, and the evidence is, the plasmids have their own DNA, same with mitochondria. They have multiple membranes. They have at least two. Cool thing, when you get into the algae, you'll actually find some plastids that are more than two membranes. So we may have multiple events where the endo-symbiont situation has happened more than once in the past. They do replicate independently of their hosts. So that's another piece of evidence that's really cool. And I can't remember exactly the reason for this, but there's a Mixotricha paradoxa has a really interesting set of um, uh, endosymbionts that really illustrate these things. So if you want to look up this, you can. It's not that important right now, but don't worry about it too much. But there's a really cool situation with them. They do retain their genes for respiration and photosynthesis. I mean, if they didn't, they would be not all that useful. They contain multiple copies of genes in the organelle. So um, they don't exhibit Mendelian segregation. So when we talk about Mendelian genetics and the way that we get like how genes are passed on, that's not the way it works for bacteria and not the way it works for mitochondria and chloroplasts. Really cool. Sperm very rarely carry mitochondria or chloroplasts. So where do these animals and plants get their mitochondria? They get it through maternal inheritance. So let me ask you, where did you get your mitochondria? Should be an easy one. Yeah, came from your mom. Okay, definitely not from your dog. Okay, didn't come from dad. So let me ask you this. If that's true, can you track maternal inheritance? Like, can you figure out who's the mom of a person just from their mitochondrial DNA? Yeah, you can. It's really cool. And we do this with plants. When I was an undergrad, we actually worked with DNA sequencing, and um, we would compare mitochondrial genes with other plants and tried to see like what's the relationship maternally through using those. So very cool. There are some weird situations where mitochondria has gotten passed in other ways. Like I said, there is at least one situation where mitochondria seems to have been passed from one plant to another, completely different species. That's really cool. So there are some interesting things out there. Okay, let's end that one and pull up the next set of slides. And here is the plant that I was telling y'all about. So we've talked about the corpse lily. Here is your corpse flower. The scientific name is Amorphophallus titanum. I will not, okay, we'll do it this way. Let me get my chat. So I'm not going to explain this name, but that genus name right there means something interesting. It really makes you wonder what was on these people's minds when they looked it up. So feel free to look that up, giggle if you want, but it does really make you wonder about the person who named these plants. So this is a corpse lily. So it's not the largest flower. Rafflesia, the one we've already looked at, is that. So the corpse, sorry, that's the corpse lily. This is the corpse flower. Maybe it's the other way around. I think they get interchangeable. But this one is the largest inflorescence in the world. I have seen one of these, and they're huge. They also stink to high heavens if you get there at the right time. The one I got to see in person that was flowering, and they don't flower every year, was not as stinky when I got there. So we had one on campus when I was a graduate student, kind of on, he was borrowed from someone. I don't know exactly how that worked. Like I said, I had some little ones growing, but they never flowered, and I don't think they survived. Greenhouse gets rough sometimes in the summer. Okay. So why is photosynthesis important? Okay, photosynthesis is important, and we're going to get into kind of basics first, and then we'll get into some details of photosynthesis. 90% of human energy that we use is derived from oil and coal. So why would I bring up oil and coal as photosynthesis important? Because they're not plants, are they? They're definitely not plants. So why would I make the connection to oil and coal? Does anybody know the origin of where we get oil and coal? Nobody?
Comes from the ground, right? Or down.
It does come from the ground. How did it get in the ground?
I don't know.
You don't know? That's okay. Anybody else know where it comes from? It does have something to do with carbon. And in fact, you're right, Haley, it is dead organisms. In this case, most likely plants, less likely animals. So sometime in the past, a large number of plants were put under pressure, and they went through a process. They died, of course, and then they were put under pressure in some other situations that created oil and coal. So where did that carbon that's in these things come from? It actually came from a product of photosynthesis. So if you think about it, most of the energy that we do on this planet right now is the product of photosynthesis, either directly or in this case, indirectly. So plants which did photosynthesis that gathered energy from the sun, that used carbon to help store it through bonds of different molecules. When they died, they didn't decompose. They went through the process of creating oil and coal. So does our planet really need photosynthesis to continue? Yeah, we do. We need it. It's really important. Okay, if you look at the amount of green organisms on the planet, 94% of that dry weight comes from photosynthetic product. It uses the carbon dioxide in the atmosphere by absorbing it and taking that carbon and incorporating it into its being, into its molecules. It is the sole principal means of sustaining life. If we didn't have photosynthetic organisms, the best we probably would have in terms of life on this planet would be bacteria. We wouldn't probably have the opportunity to be high level eukaryotes on this planet. Now, one thing I want to really point out is that we do not make energy. And I may say that from time to time. It's not because I mean to, it's just because we slip in the way that we can converse about this. We do not make energy. You know, the laws of thermodynamics do not allow us to claim that we can make energy. You can transfer energy, okay? And we transfer it from bonds to bonds through chemical processes, okay? So enzymes are used as part of these chemical processes that regulate metabolic activity. So we know that there's anabolism, where it stores energy. Photosynthesis is all about anabolism. It's about building things, storing energy in multiple new bonds and larger molecules. Catabolism, on the other hand, is consuming stored energy, breaking bonds, releasing that energy, and this is respiration. And of course, we use both of these in this planet to kind of keep energy moving. This is done through a bunch of oxidation reduction reactions. Redox sometimes is how it is abbreviated. Oxidation is the loss of electrons and reduction is the gain of electrons, and they're usually coupled together, so you get an oxidation and a reduction. Oxidation and reduction. By the way, how many of y'all have had chemistry? Okay. Did y'all learn any little acronym for how to remember loss of electrons is oxidation. Anybody have Dr. Roy? I know he usually tells y'all oil rig. Does that sound familiar? Oil rig? Okay. Oil rig is oxidation is loss. Okay. And reduction is gain. I don't like oil rig personally. I learned Leo says GER. Loss of electrons is so it's loss of electrons is oxidation. Gain of electrons is reduction. That's me personally. I just like the idea of Leo the lion. That's how I remember it. And I accidentally sent that only to Brandy. Let me do that again. There we go. Leo says GER. Crazy enough, I didn't learn that until my last chemistry class. And since I had a chemistry major, that's impressive that no one taught me that beforehand. Okay. So what does photosynthesis use? It uses carbon dioxide from the atmosphere. And carbon dioxide reaches chloroplasts in the mesophyll cells. So there's these cells, they absorb through holes called stomata in the outer, usually under portion of the leaf. So it has to get into the leaf to be able to be used. Water is about less than 1% that is absorbed. Most of the that's used, most of it is actually either transpired out of the plant, which means that the water it goes up to the plant, but it ends up leaving the leaves into the atmosphere, or it becomes part of the plant. It doesn't get really used in photosynthesis as much. And 80% of the light that plants have available is absorbed and used. Now, can anybody tell me what color of light is not used by plants? It's going to be green. Okay. So how do we know just by looking at plants that green is not used? Because they are green. Exactly. So what's happening with green light when it hits a plant? It's being reflected. It can also be transmitted. So it can be transmitted through the plant. So if you've got a thin enough leaf, you can hold it up and you'll see green come through. But for the most part, it's being reflected. We see it because it's reflected back to our eyes. The other colors are absorbed. Okay. So good to go there. Sounds like y'all have that one pretty well in hand. Okay, so let's look at some effects on photosynthesis. Elena, you probably will recognize these. These came right out of Plant Viz. So plants, there are a couple of kinds of photosynthesis. We'll talk about those. There's C3 and C4. There's actually 3 kinds. Notice that as you increase light intensity, you get an increase in both of your photosynthesis kinds. But there is a point at which one of these can handle more intense light, and that's going to be your C4 plants. C3 plants kind of level off, and at some point having more light is not helpful. On the other hand, if you look at temperature, as you increase temperature, C4 plants are just fine. C3 plants decrease. Okay. So real quick, do you see any implications with global warming in the future? So what do we see what's happening to the planet? Is it getting warmer? And
Yes.
do we notice that right now the weather's getting strange too, isn't it? A little bit. I'm a little concerned. Did anybody hear the other day that Greenland had rain for like the second time in centuries, maybe? I don't know. I don't know the last time. So here's the problem with rain in Greenland. If it's warm enough to rain in Greenland and not snow, it's warm enough to melt stuff. And does anybody know what happens when um, rainwater hits ice? I know most of y'all are from Mississippi. You don't even know what ice is. It just comes out of the refrigerator, right? Have y'all ever been places where it's like regularly you get ice on the road? Okay, so where I grew up in Tennessee, because we're kind of that mid-range, we could get ice, we could get snow, we could get freezing rain, we could get regular rain. And anytime it was warm enough to get fully like into regular rain, even though it had just iced maybe overnight, what happens is it will melt that ice that it hits. Because if you think about it, if it's a liquid water, it's warmer than the ice, right? So it would melt the ice. Now, overnight, it'll freeze again. And so in Greenland, yeah, they're going to freeze. That rain is going to freeze. But here's a problem. Just little side note on this. Frozen water is different than compacted snow in its ability to reflect the sun's rays. So as Greenland melts, it's going to make the Earth warmer. We're losing one of our reflective shields. As it's warm enough to melt the ice there with rain, even though it may become ice, liquid ice, think about it. An ice cube, you can see through it, can't you? It doesn't reflect. But if it's cold enough to be snow instead of ice when it comes down, it compacts into a big white blanket. I'm expecting things to get worse, just a side note. And that's what plants are going to have to deal with. Now, notice what happens as temperature increases. There are a group of plants that don't do as well, right? And this should tell us something. We've mentioned that photosynthesis uses enzymes. Are enzymes temperature dependent? Can you affect an enzyme by changing its temperature? Yeah, you can increase the rate of reaction. You can decrease the rate of reaction if you cool it down. And what happens if you overheat an enzyme? Does it go into crazy hyperactive mode or something else happen? There's a special word I'm looking for. Starts with a D. Any ideas? Denature. Yeah, exactly. So what happens to an enzyme that denatures? It actually unravels. It's gotten so hot that it loses its three-dimensional structure and it's not able to function. We don't want that. We definitely don't want our photosynthesis to stop working. Now you might say, oh great, we'll just use a bunch of C3 or C4 plants instead. Well, there's a limit to which they can handle this as well. Okay, they can go into higher temperatures and be okay. But notice this graph only goes to about 40 degrees Celsius. If you have any idea, 40 degrees Celsius is just over 100. We really don't want to go higher than that. And by the way, does anybody want to live in a place where it's like over 100 degrees all the time? No, not at all. Not even part of the year. Like I'm dying over here with humidity and like 90 something degrees. I don't know. I just don't do well with humidity. We just don't want to deal with this. Plants can't deal with it up to a certain point. Some people are like, oh, great. Plants absorb carbon dioxide. The more we put it in the atmosphere, the better it is for plants, right? Well, that's very simple thinking. And the same people that say that are also people who say that this SARS-CoV-2 virus is small enough that it can pass through um, your mask. Both situations are technically correct. Yes, plants like carbon dioxide. Yes, the virus is small enough to pass through the mask. The difference is that too much carbon dioxide is a bad thing, and virus does not travel by itself alone. It's usually a part of a liquid droplet. So the mask can catch the liquid droplet. We're not looking at virus particles. We're looking at virus particles attached to something. Y'all see what we're getting at here? A little bit of knowledge can be a lot of trouble, okay? And we're trying in our classes to help y'all have a better, bigger picture understanding of all these things. I tell you, it worries me the amount of people out there that think they know what they're talking about. Sometimes I'm one of them, to be truthful. I find that I assume a lot of stuff, and I need to be more open to taking in new information better. So I need to also pay attention. I think about myself as well. It's always good to put yourself in everybody else's shoes. Makes you more understanding. Okay. Got on the soapbox there, guys. Sorry. Okay, we use the carbon dioxide. We talked about this. I went the wrong direction, I think. There we go. So light that's too intense is definitely a problem, and it can accelerate photorespiration. And it's a process where you can produce some energy, but it's wasteful. It uses oxygen and it releases carbon dioxide, and that's not how photosynthesis works. It may keep the plant alive for a short period of time, but it's not something that can continually be done. Photorespiration is only used in situations where regular photosynthesis is not working, okay? Imagine a plant getting overly heated in the greenhouse. It might switch over to photorespiration just to stay alive and not lose all its water. You can also have photooxidation. This will actually destroy the chlorophyll. The light will overwhelm and destroy the chlorophyll molecule. That's definitely a bad thing. And also you get the induction of color change through a process of photooxidation. So in the fall, as plants are getting ready to lose their leaves here in this part of the world, not true all over the world, but for here we have color change. And a lot of times what happens is those chlorophyll will break down, and you'll start to see the colors in behind the chlorophyll, the xanthophylls, the anthocyanins, or whatever the case may be. Okay, chlorophyll A, important. All plants and algae have chlorophyll A, as far as I know. You want to make this as a note. We will see it again. I will ask about chlorophyll A. It is extremely important for photosynthesis to take place. There's like 3 times more chlorophyll A than B. So if I ask you what type of chlorophyll is most important, or what pigment is most important, can you just say chlorophyll, or should you say chlorophyll A? You know the answer, right? Do chlorophyll A. Try not to leave that off. Chlorophyll B is considered to be an accessory pigment, and it's yellow-green. And what it does is it absorbs in the yellow and green. It absorbs outside of the yellow and green range and transfers that energy to chlorophyll A. So chlorophyll A. This is one of those things that bind all photosynthesis together. There are some other pigments outside the chlorophylls. There are the carotenoids. They are yellow-orange. Guess what colors they don't absorb? In the yellow-orange range. That's why they're that color. We don't usually see them because they're masked by the chlorophyll, because there is so much more chlorophyll A. That's what you see, is that green color, because there's more of it. But when in the fall the leaves start to change and the chlorophyll breaks down, you get to see some of these other colors. Phycobilins, these are blue or red, and they're found in cyanobacteria and red algae. There are, of course, the other chlorophylls we mentioned, and there are also photosynthetic pigments in bacteria that are not found in plants, but does allow bacteria to use light to produce energy. Here's a little table I found in one of my textbooks, and it's really nice. This little visual diagram of what I just said. Okay, I'm not going to ask you specific things off of this diagram, but can you tell me something? When you look at protists and plants, what's the biggest difference in terms of photosynthesis and pigments? Yeah, Elena, I thought I showed y'all this one. I thought I did.
I think you did. I just can't remember.
This one's actually out of a. By the way, she was asking me if we showed this in plant physiology. It's like I think I did, or one like it. This one actually is out of a general biology textbook. Well, what group of pigments do protists have that plants don't? It's the phycobilins, yeah. So what that tells us is that the plants probably developed, and there are other reasons why we think this too, developed in the protist group. So plants came out of protists, which is not all surprising because algae falls within protists. And while algae are not considered to be plants, algae are very similar to plants. They're just usually not multicellular. They can be, but not to the same degree. We'll talk about that too as we move along. So probably what you notice is that basically plants have a subset of what is found in the protists. And that's kind of an evidence for us to see the development from protists into the plant kingdom. Okay, I'm going to stop right there because I've got like 1 minute left, and I think that is plenty for us. I think it'll make more sense to stop at one spot. Where did it go? Yeah, we'll get directly into photosynthesis, slide 10 on Monday. Make sure that you get your lab handouts printed out, or on a computer or some other way, however you're going to bring them, just bring them that way. I would suggest, especially if you're using your book on your computer, you might want to have your handouts on paper so that you can look at one and have the other. I don't know about you, I get annoyed having to switch pages on the same computer. Now, you may be able to pull them side by side. That's a possibility, but just I think it's useful to have it on paper, but that's just me. Okay. Any questions before we go? We'll be back on Zoom on Monday morning and then live in person.
I have a question about the quiz.
Yes.
I know the short answer and stuff. Are they? I'm trying to word my question. Are they like detailed questions or just kind of straightforward or mixed? I was just kind of curious.
Probably going to be mixed. I'll probably have
Okay.
to define some things on the easy side, but I probably will have you describe some processes. Like I may have you describe to me photosynthesis, but only as detailed as we might go in class. So, I mean,
Okay.
I wouldn't necessarily ask you something that you wouldn't know in class. Does that make sense?
Okay. Yes, sir.
And it may not be to the same detail that we cover, but what I would do is I would prepare to be able to explain any processes and then
Okay.
to explain or describe any sets of terms. Okay.
All right.
Elena?
Is that where you're talking about drawing the processes, or do you want us to like actually draw like something?
I'm trying to think if there's anything that I would want you to draw. The only thing I could think of right now would be a plant cell to illustrate the maybe major components that are plant specific, maybe over what's also found in animals.
Are we allowed to draw the process, or?
Okay, I will tell you, sometimes I will ask for drawings, And then there are other times where I'll ask you to answer the question, and if a drawing is the best way to answer it, and I can get what you're getting out of it, like I can tell you know it, it's not just, you know, repeating a drawing, I'm fine with that. That's me. If you can answer something with a drawing, sometimes better than with writing, I would much prefer to have it. One, it's easier for me to grade a lot of times, and two, I think you can illustrate more understanding in drawings a lot of times, not always, but a lot of times. So yeah. And like I said, this will be probably a Word document that I'll have you just fill in your answers to. I mean, I can make it a Canvas. Would y'all want me to make it a Canvas quiz where you type your answers into Canvas? And then so I can make questions where you upload pictures.
I would rather the Word document.
Okay. Okay. Anybody else weigh in on that? Do you care one way or the other?
I like the Word document.
Okay. I mean, it's just as easy for me to grade it that way. I can do it in SpeedGrader, and then I can do comments out to the side. So it'll just be like you took it in class. Okay.
One more question, sorry. When will it be posted for us to print?
I can't do it early because then you would.
Yeah,
Do you have these?
because my only problem is I would have to go somewhere to print it. So
Well, what about this? Can you just do it in like answer anything you type into the document? You got to have a picture, draw it on something else, take a picture, and then upload that as a separate file.
okay, yes, sir.
Yeah, because Canvas will let you do that. You can upload multiple files. And what I'll do is we're going to test this out. It may take the whole time on Wednesday to take this quiz, because we won't. I mean, I'm not going to make it more than 10 to 12 questions, and 12 only because probably there's some bonus. Okay, but I'm not sure how this is going to slow us down. If it's in person, it's fast, but with technology, we slow down. We'll see how it goes, and then we'll go from there. If you have any more questions, we'll cover it on Monday, and we'll take it on Wednesday. Okay, and the quiz will cover through whatever we finish on Monday in photosynthesis. Okay. Well, y'all have a good weekend. I will see you later, and I'll get these videos posted too. Okay. Okay. See you later. Have a good.
Thank you.
Welcome.